It's very nice to be here. I'm very grateful for the invitation, and I was just saying to some colleagues earlier, I've come from Edinburgh overnight, and so if I fall off the stage and dislocate my shoulder like David Forbes, I'm hoping those in the front row will assist just as you promised to help him. I'd also like to thank Tony Dell, Miriam Dwyer and Andrew Koo for the opportunity to be here and for all the hard work they've put into this magnificent event, and to thank RSL Queensland for funding the work that I'm going to describe to you at the end of this talk, and to my colleagues at the Gallipoli Medical Research Foundation, particularly Professor Bruce Lawford. He and I have been working together for some time. As a clinical psychologist, I've been interested in the strengths and the limitations of the work that falls into my professional domain. And some of the challenges are those that you've heard about earlier today. It's about how we integrate bodies of work that come from different disciplines to get a more coherent and meaningful whole. And I think in PTSD, that's a particular challenge. It's a challenge that I think is timely to face. There's increased public attention around PTSD. This is a recent popular scientific magazine called Cosmos that featured an integration of some of the current work on PTSD, including epigenetic research that I'll talk about today. And I think it's a broader coalescing of commitment to serve those who have served us with better science to help to understand the consequences of their service. And as you've heard earlier today as well, we've got good estimates of some of the consequences of service in terms of mental health. The Brian O'Toole study that was mentioned earlier has a lifetime PTSD prevalence estimate of about 21% in Australia, and that international review that was also mentioned earlier today has estimates in first responders of 4.7 to 14.6%, with ambulance officers having the highest prevalence. Just from the outset, I want to be clear that PTSD is complex and there's a range of different factors that contribute to the disorder, but I'm here to talk about genetics today and acknowledging the array of other factors that may be biological, non-genetic factors, psychological, social and cultural factors that can contribute as well. But in essence, PTSD is a quintessential biopsychosocial disorder. In psychiatry and psychology, that's the kind of model, the frame of reference that we typically use. But in DSM, it's the only disorder that has a sharp and demarcated precipitating event, a criterion A event, one that where you feel as if your life is under threat, or indeed it is under threat. And so it's a particularly important disorder for us to understand, looking at how that event relates to other risk factors to produce the disorder that we're talking about today. And that risk has been acknowledged to be genetic in part for some time. We've talked about World War I as part of the genesis of some serious attention that was paid to the resultant mental health consequences of combat. And indeed, this early paper, which was in The Lancet in 1918, talked about a family history of psychoneurosis in those veterans who had suffered this disorder. And when we try and estimate the kind of genetic underpinnings of risk for liability and symptoms, various studies come to estimates of somewhere between 30 to 46% for males, and it's thought to be considerably higher for females. So what I'm going to do this afternoon is to talk about some of the work that we've undertaken over the last 15 years. And I'm going to begin with a review that Joe Voisey was the lead author in last year, where we've summarised the best to the best of the extent that we can, all of the genetic work that we've seen in PTSD and see what kind of themes emerge. But inevitably, as part of that, there's going to be some scary figures, and some scientific terminology, which sadly is unavoidable, but I'll try and just illustrate the points as best I can. There's a couple of key themes that I really want to emphasise. The essence of some of the scientific detail really isn't that important. This body of work that we referred to last year in a review article looked at 68 candidate gene association studies. So these are studies that look at a particular gene and then look at the association between that gene and the diagnosis of post-traumatic stress disorder. In addition, we also included 6 genome-wide association studies. So they're studies that essentially look at all genes and then look at a large number of genes and identify differences between those with the clinical condition and those without. And we also looked at 17 epigenetic studies, and these epigenetic studies look at how genes are modified and their effects to produce different results. And those 17 studies had looked at 48 genes, and there were also a GWAS study there too. When I've divvied up this work into whether it occurred early or later and the nature of the trauma that's been looked at, you can see there hasn't been a conspicuous growth in genetic studies over recent years that look at combat, and I think that's a very important issue for us to address. So over recent years, there's been work that looks at things like urban trauma and stress, so being reared in a violent community, one that's very conflicted, for example, and there's also been recent work that's looked at genocide, for example, victims of the Rwanda conflict. And in all this body of work, there are no studies that we could find that looked at first responders. I think it's very important for us to focus on those studies that looked at combat trauma. Arguably, the combat trauma could be different. It's likely to be repeated exposure, and there may be a cumulative effect in terms of a dose effect of that repeated exposure as well. So I've extracted the studies that we looked at that look at a range of genes, and these are the list of the genes names here on the left, so it doesn't really matter what they're called. Essentially, these are genes that are associated typically with hormones like stress hormones or brain chemical neurotransmitter functions. And those that are highlighted in yellow are those that weren't significant in terms of the studies. So when we're looking at this body of work, there's a couple of things that really leap out. One is when we're examining these combat-related studies, the numbers are quite small, but secondly, replication is rare. So many of these published results have just looked at these gene associations once and then not followed them up with other studies again. The first of those genes listed, the ANC1, which is a gene that's associated with the brain chemical dopamine and its function, has been looked at most often, and most often by the team that I'm part of here in Brisbane. So we'll talk about that in a minute in a little more detail. But before I do that, I'm going to refer to one of the first studies that we did where we'd hoped to look at genetic associations. And there was a bit of a challenge because when we asked participants were they willing to provide samples for DNA assessment, the majority of them said no. And I think we've built good trust over the time, particularly at the Keith Payne unit, where we've had very strong and willing engagement from participants. But I think when we began this genetic work, there was a degree of suspicion around the role that genes may have in PTSD. And in fact, many of the veterans in this first study were concerned because we were sending the samples to the United States for analysis and just didn't like the loss of control in terms of what may subsequently happen with that investigation with our collaborators at UCLA. So although this first study, which was a subset, was going to be looking at genetics, it was part of a national study where we had 608 participants who were in trauma-focused CBT from across Australia, and we were interested in the relationship between alcohol misuse and post-traumatic stress disorder, given the common comorbidity that we see there. Essentially, what we found, and this is without the genetic analysis, was that alcohol use at baseline for these patients was not predictive of their PTSD outcome. In fact, there were those that became low-risk drinkers by fully engaging in the alcohol-related therapy as well as the PTSD treatment, and at 9 months showed less avoidance, numbing, and arousal compared with those who hadn't changed their drinking behaviour. And we saw that when these improvements were early in terms of drinking, there were greater changes in PTSD symptoms post-program, so in terms of natural exposure to the environment. And we've heard today about how exposure is an important mediator of therapeutic change, and alcohol may buffer the benefits of that exposure and ameliorate or minimise it. So removing alcohol out of the picture was clearly important. But not everyone can change their drinking that easily. And so the next study that we did, we were very keen to see if there was a genetic contribution to this effect, and indeed there was. So we looked at the gene that I've already mentioned, the ANK1 gene, which relates to dopamine receptors in the brain, particularly the density of D2 dopamine receptors, which are related to reward. So dopamine's an important neurochemical that signals reward in our environment. And we also had identified this gene and examined this gene in substance misuse in a number of other samples. So in this study, we recruited 91 male Vietnam veterans. We asked them about their drinking behaviour in Vietnam, and all reported exceeding 60 grams of alcohol per day when off duty during their service. But at the time of the study, 41.8% exceeded that cutoff for heavy drinking at 60 grams, and 21.3% were abstainers. What we found when we separated the group into those with the risky version of the gene associated with pathology, so that's the A1 version, A1 plus, and the A1 minus, is that the average amount of alcohol consumed per day by those with the A1 plus status was about 140 grams. That's 14 standard drinks, versus those without this allele, where the average was about 60 grams. And consistent with the notion that this may be self-medication for PTSD, we also looked at the rate of consumption, and there were striking differences. So this is the rate of consumption per hour on average if you have the risky version of this gene, which is over 50 grams of alcohol. That's about 5.5 standard drinks per hour, and about half that for those without this risky allele. So we were intrigued by this. We were keen to explore more fully as to why a single gene associated with a chemical in the brain may be related to such heavy alcohol use. And so we followed that up in several studies, and I'll just mention a couple of them here today. The first working hypothesis that we had was maybe there's a more severe form of PTSD amongst this group. And so this is a technique called cluster analysis, where we used the General Health Questionnaire 28, which asks about psychological symptoms, and then grouped them into the participants into 4 clusters on the basis of these symptoms. And this is actually just comparing the Mississippi scores. So this is a measure of post-traumatic stress disorder symptomatology amongst these 4 clusters. But we also looked at the 4 GHQ symptoms as well. Essentially, we found a severe pathology group. So this is those with the highest PTSD, and they also had the highest General Health Questionnaire symptoms as well. And within this cluster, there were 12 A1 positive individuals and only 4 A1 minus. In this cluster here, that proportion was reversed. So you had 24 A1 minus in this low symptom cluster and 8 A1 plus. So our hypothesis that maybe this conferred a more general risk for PTSD complexity or severity, and that wasn't attributed to alcohol, we portioned out the effects of alcohol in the study, was borne out. And we then began to think maybe this group would also show differential outcomes in terms of treatment. And we examined the treatment that's been mentioned today, the most successful treatment, pharmacotherapy treatment, which is SSRI antidepressants, and we chose to run an open-label clinical trial using the drug paroxetine. And we selected this drug paroxetine because we know that when you take paroxetine, it increases the density of those D2 dopamine receptors I referred to before. So it's an active drug that may compensate for the deficit that we see in those A1 plus individuals who tend to have lower numbers of dopamine receptors. There was 63 veterans originally recruited for the trial, but 18 discontinued. And interestingly, there was a trend for those who dropped out of the study who had the less risky version of the gene because they experienced more adverse events. And we think that was probably because it produced too many of these dopamine receptors in the brain, and that became aversive. But that notion of personalised medicine and trying to match the individual on the basis of their genetic constitution to a particular drug is an important one. Look, this slide's not particularly vital apart from this bit here. This is one of the 4 General Health Questionnaire factors measuring social dysfunction. And what we saw here was there were significant effects in terms of treatment for depression, so depression got better, for anxiety and insomnia, that got better on paroxetine, and so did social dysfunction. But the only difference here was really amongst this A1 plus risky group. And we thought this was a potentially important finding, that on the basis of increasing the number of these receptors in the brain, that individuals reported less social disability. That meant that they could maybe engage more fully in group therapy, more actively seek social support, and maybe more fully interact with their families. So once again, it became an interesting piece of a jigsaw looking at just one gene that impacted in a number of facets of PTSD. And then we also then examined other related polymorphisms or parts of this gene related to dopamine to see if they were also significant too. And what we found was that there was an overall effect here for this C957T gene, which actually explained the effect of the gene that we looked at previously. And we quite often see that because genes that are close to each other in a chromosome can influence the expression of another gene. So this gene wasn't significant, and the gene that we looked at was actually explained by an adjacent gene. But what we could see from that, though, was that a dopamine-related gene was associated with PTSD diagnosis. We've then moved on to look at some specific genes related to a particular part of the brain. We heard this morning about the ventromedial prefrontal cortex and the amygdala and their importance in fear conditioning. There's also another important structure of the brain, the hippocampus, that relates to memory function and the integration of sensory input into memories. And we've also been interested in looking at genes that exist either in animals or in humans that relate to hippocampal function. And the first one of these is related to despindin. And despindin helps to regulate an excitatory transmitter in the brain called glutamate. And we know that when glutamate changes, there's also changes in sensory perception. So we were very interested to see if this gene that relates to something that influences your perception of what's around you in terms of what you see and what you hear, and influences a structure in the brain we know associated to be associated with PTSD, if this would also be important. But what we've done in this study that was different to other studies, we were keen to see if this was a broad risk just for PTSD or if it related to mental and ill health in general. And that's what we saw. We saw a significant effect with all of these disorders: schizophrenia, PTSD, nicotine dependence, and opiate dependence, but not in this study with alcohol dependence. And so I guess there's a salutary lesson there too about the fact that we might have broad ill health that relates to some of these genetic factors, and that might be useful, but they won't be particularly good biomarkers for PTSD on its own. And the second gene that we've looked at that relates to hippocampal function is the NOS1AP. And this is a gene that regulates the dendrites in nerves. These are the enriched connections that connect one nerve cell to another and are related to signaling of this neurotransmitter and ultimately hippocampal volume loss, so a smaller hippocampus, which we know is a risk factor for PTSD and is probably also a consequence of traumatic exposure and PTSD as well. So in this study, we saw that having both G alleles of this NOS1AP gene, in this one we examined 13 different varieties within this gene, but there was one in particular that showed a 3 times, so this is the odds ratio, so a 3.2 fold increase for the GG genotype versus not having any Gs in your genotype, having the AA genotype. And importantly, we also saw that this genotype was associated with higher scores on the Beck Depression Inventory, where there's also been previous studies that have looked at this NOS1AP and depression, and also with the Mississippi scale score, the scale I talked about before that measures the severity of PTSD symptoms. So bringing this together, just looking at some association studies of single genes, or sometimes more than one gene, and sometimes with more than one disorder, there's some broad conclusions to draw. First of all, on the basis of our review, while the body of work on the genetics of PTSD appears to be growing, the proportion of work that's dedicated to those who have combat-related exposure isn't, and we're doing our best to try and advance this work. That looking at individual candidate genes, these studies are rarely replicated, and that's vital. The essence of all science is replication. The ANK1 gene is the most studied to date, and most studied to date indeed by our team, and it doesn't appear to be a marker for PTSD. There's been controversy within the literature about that, but our conclusion is that it's associated with more severe symptoms, with heavier self-medication of alcohol, and an enhanced treatment response to paroxetine and SSRI. But our recent studies examining genes that affect hippocampal functioning, a structure of the brain related to memory, appear to be more promising. But one of the issues that's really struck us as we've progressed this body of work has been the parallel of genetic technology, that we can now look at multiple genes at the same time. So there are GWAS studies that have done that, but there are also chips that help us to do that work as well. And there's been an increasing realisation in the body of work as a whole that no single gene alone is likely to be of primary value as a marker of PTSD. This is a complex disorder where it's likely there'll be a complex interaction of genes. Or, in addition to a complex interaction of genes, there may well be epigenetic effects as well. So the second part of this talk's really just to introduce you to the concept of epigenetics. And this is a recent field in terms of its molecular biological understandings. It's emerged out of some very important twin studies which have largely been conducted in Spain that show that identical twins can have very different risks for disease. And while epigenetics used to be considered as the interaction between genes and environment to bring a certain phenotype or the expression of your genes into being, it's now considered to be the additional instructions that influence gene activity in your chromosomes, but without altering the sequence in your DNA. And one of these milestone studies, which I won't talk about in any detail, that occurred in Spain, was just looking at as these identical twins age and they're exposed to different environments, the expression of their genes changed as well. There was an adaptive interaction between genes and environment that produced very different biological results, including things like risk for cancer. And this occurs through two primary processes. And I won't describe the processes in much detail, but they're illustrated on this slide. The first is called methylation. And methylation's when the DNA is altered by methyl groups and essentially influences how that DNA expresses itself to produce proteins ultimately. And the second's more complex, and it's called histone modification. And histones are proteins that package DNA and into something called chromatin. And those histones can be tightened or untightened. And if they're tightly packed, gene expression is shut down or silenced. But we know that environmental exposure can influence both methylation and the expression of genes in this manner, or the open or closed nature of histones through histone modification. And so last year we were very keen to progress this body of work, and work that Joe Voisey led, looked at, we were the first group in the world to examine gene expression in brain tissue in schizophrenia. And I've offered this as an example. We haven't done this work yet with PTSD because we were actually unable to find in brain banks in the United States brain banks from those brains who have been donated to brain banks from those with PTSD. But this work with schizophrenia took post-mortem samples of about 0.4 or 1 gram of frontal brain tissue in the regions that we were talking about this morning, and uses this gene chip, which allows us to look at 485,000 sites simultaneously. So it has amazing scientific power. And the analytic tools that we have allow us to do this with very small samples. So this is robust scientifically because of the underlying statistical theory that drives it. And when we adjusted for the age, because we had a significantly different age between the groups with schizophrenia and our non-disease controls, and post-mortem interval, which is the delay between death and the brain being preserved, we could see that there were 4,641 probes that were differentially methylated from 2,929 genes. And we undertook a cluster analysis, just as we did in that previous study, but instead of looking at symptoms this time, we looked at methylation. And interestingly, we found genes that are similar to the genes that we've looked at, or identical to the genes that we've looked at previously. But what this shows is that it's the mechanism that the genes are using to produce outcomes related to dopamine, the transport of neurotransmitters, or the actions of dysbindin. And so we believe that this methylation, which can be switched on or off through environmental events, is likely to be crucial in certain developmental periods for the development of schizophrenia, probably in schizophrenia, particularly in utero. And the epigenetic events that might affect methylation here could be events such as exposure to an influenza virus, because we know that the diagnosis of schizophrenia likelihood varies according to whether there's an infective epidemic during pregnancy. And this is just related to the 6 primary genes that we've looked at, just examining their difference between controls and those with schizophrenia. But we're very keen to do similar work in PTSD. Other teams have, and some of the work that's impressed me most has been with the Dutch Armed Forces. This is looking at glucocorticoid stress-type hormone activities and a glucocorticoid receptor inhibitor called FK506, and the activity of the messenger RNA or methylation of this gene is also associated with PTSD symptoms. And importantly, demethylation of this DNA from the same gene has been found to mediate the interaction between exposure to childhood trauma and whether or not when you're exposed to trauma in adulthood, you're going to develop post-traumatic sequelae. So that's a very important finding that links early exposure to trauma, later exposure to trauma, and psychiatric disease using a particular epigenetic effect with this FKBP5. There's also a very important recent study that's just come out this year, a British collaboration, but it's looking at inflammatory markers. And these are interleukins. We spoke about them just previously in Professor Crawford's talk. But it's looking at hypermethylation, so that's increased methylation of some of these genes, and then undermethylation or demethylation of other of these genes, and 188 US Marines. And one of the important features of this study that many studies don't have is that they had data from pre- and post-deployment relating to RNA expression, and then used this very complicated network analysis that helps to link up the genes and the gene products in a diagram that's absolutely astounding. So when you see the next diagram, don't be too overwhelmed because I don't possibly understand it either. But essentially what these diagrams show is that when you looked at what these networks mean of these genes that are either under or overexpressed in US Marines pre- and post-deployment, is that they relate to immunity, and particularly wound healing. And so the working hypothesis from this work that's looked at this network of all of these genes and gene products and then looked at the reference standards that this relates to, these networks are related to the body's capacity to heal itself. And so that's a really important epigenetic finding that has broad implications. When we think about the future then in terms of genes that might influence our stress hormones, switching on or off in relation to early childhood exposure, or whether combat exposure and whether those genes that switch on or off relate to our immunity, there's significant scope for epigenetic work. And as we've heard several times today too, that large-scale studies are more powerful and more robust in this regard. And there are some large-scale studies occurring now. There's the International Human Epigenome Consortium that follows on from the ENCODE project in 2012, which used human cell lines as opposed to looking at clinical cases. And the NIH project that's undertaking, there's 127 human tissues that are being analysed in terms of their epigenetics to provide a comprehensive epigenome encyclopedia, essentially a reference work that we can gauge our studies and other disorders such as PTSD with this large-scale reference work. And then there's also the Psychiatric Genomics Consortium that we've also heard about earlier today with 40,000 cases, 40,000 subjects rather, 10,000 cases and 30,000 controls. And the Human Epigenome Project will be able to eventually, so it's using tissues, a variety of tissues. This just represents diagrammatically the kind of tissues and cell types they're using. And you'll get diagrams like this that relate genes to particular functions and that the width of these pathways relates to the strength of activity. But this will be able to provide a very integrated and connected view between genes and gene products and biological functions. So if I was going to think in a similar way to the other talks we've had today about how can we advance this work that looks at the complexity of genes, the interaction of gene and environment that influences how these genes express themselves, we certainly need to compare those who have been exposed to trauma and have PTSD with trauma-exposed controls. We need to be able to look both at PTSD and at other distress that doesn't represent PTSD. And importantly, we've heard from several speakers to move beyond the diagnosis, to examine how this relates to symptoms and indeed functional outcomes as well. The complexity of multiple genes, their expression, and of environmental influences all need replication. And ideally, as we've also had underscored by several other speakers, the only way to truly understand this is to look at the unfolding events over time and solid information pre-deployment, biological information, as well as social and psychological information is vital to the progress here. And just as an aside, well, not really an aside, in fact, as a central aspect of this, we need to consider the ethical issues of engaging in this work from the outset, because there are issues related to both lowering stigma and enhancing stigma, of reducing something complex in popular views to something that's very simple and reductionistic, and to have a whole range of issues in terms of discrimination from health insurance to employment that might be a consequence. So the study that you've just had described to you, which as you saw I'm a chief investigator on, along with Professor Crawford, initially sought to recruit 300 Vietnam veterans, and we're going to be recruiting those that have been in the Iraq and Afghanistan conflicts from 2016, but to look at a broad array of psychological and medical parameters, of disease states, and then to examine epigenetic effects in a subset with blood. But importantly, you've heard we have semen samples, and it's really vital that we also look for epigenetic effects in those semen samples as well, because you can inherit those epigenetic changes. When I talked about that methylation and histone modification, that can be passed on via male germ cells. So we think that that's a really important and under-investigated area that we hope to advance. This is the current Illumina psycharray chip that we're using, which allows us to look at 271,000 different varieties of genes. And this has only become available last month. So we're one of a few groups in the world that's begun to use this technology very recently as well. And just to end with something that's more speculative, but I think that that's really important to consider. The healthy warrior effect was mentioned earlier. Initially, those that have PTSD were probably the healthiest people in our community, and that's not the case anymore. There's ample evidence to show that multiple physical diseases are associated with post-traumatic stress disorder. We've also heard today that sometimes I think these issues have been dismissed as conversion or somatisation or Gulf War type syndromes, and there's certainly work to suggest that others have said that the highest proportion of ill-defined physical disorders of any psychiatric condition are associated with PTSD. These physical health outcomes may well be epigenetic. They may well be related to the ongoing effects of the high levels of adrenaline, noradrenaline that we've heard about before, or other factors that may switch genes on and off. And as part of our study, with the Gallipoli Medical Research Foundation. We hope just not to examine those clusters of symptoms that we've talked about before that are psychological. We also hope to look at the interface between psychological and physical disease. So I guess in summary, I'd state that the risk for PTSD is clearly in part genetic, that recent advances in epigenetics just over the last 5 to 10 years have demonstrated resoundingly that our environment can alter gene function and that we need to bring this knowledge to the table to understand PTSD fully. That epigenetic influences can be intergenerational and they may exert themselves at crucial developmental periods, such as in the womb or in childhood or adolescence, but can express themselves throughout your life. And the manifestation of these changes may well be physical and could be mediated by the immune system. If these epigenetic effects are established, and we've heard today that something like exercise may help to ameliorate them, but I'm sure that we'll also find that things like good nutrition will help to ameliorate them too. But we also need to pose bigger questions about the kind of society that those who have served are trying to ensure that we have, one that's free and fair, one that's adaptive and health promoting, one that's less violent for everyone, including those in our Defence Force and our first responders. To not embrace those much bigger and more full challenges will mean that epigenetic effects continue to occur and manifest themselves in a number of ways that will mean that people won't fulfil their potential. I believe that in terms of our enriched understanding of PTSD, epigenetics has much to offer in the immediate future. Thank you.